Hi everyone. Uh, Very good evening on the eve of our Independence Day. Yeah, so it's nice time to meet you and give some informations. So to begin with, I'm Sridhar, who has recently joined, and I'm the senior manager for the products, who is taking care of the one products division of Genotypic Technologies. And in this meeting, I take an immense pleasure in welcoming you everyone to this webinar, faster and efficient NGS analysis using Commander NGS sequence analysis software and GTGPU HPC machines for your data analysis. So genomics has become an essential tool in every field of biology, and NGS has become a key skill to learn. We all know that it is easier to generate data than analyzing it. Having said that, Commander NGS is dreamt, designed, and developed in India by Genotypic Technologies. Commander make analysis job easier for any biologist who wish to publish NGS data of his or her research. Without complex command line scripts, one can perform QC to alignment, assembly to annotation to prepare your customized reports. On this note, I would like to introduce you to our speaker, Dr. Raja Mugasimangalam, a veteran technocrat turned entrepreneur in genomics sector. Dr. Raja Mugasimangalam is the CEO and founder of Genotypic Technology, India's first genomics company, and Qtiolomics, India's first agri-genomics company. He received his master's and PhD in biotechnology as part of the first batch of uh, national entrance test conducted by NBTE, and now it's a TVT from Madurai Kamaraj University, following which Dr. Raja had postdoctoral stints at Weizmann Institute of Science, Israel, Sydney Kimmel Cancer Research Center, and at Argonne National Lab, USA, where he developed novel methods of DNA sequencing and analysis for the Human Genome Project. Prior to founding Genotypic Technology in 1998, Dr. Raja worked at Quark Biotech Incorporation, developing gene expression assays for studying drug toxicity. Raja is well recognized amongst the scientists in academics, government, and industries alike, and has over 25 peer-reviewed publications in various national and international journals and multiple patents to his credits. So I welcome Dr. Raja Mugasimangalam to give his findings on the NGS analysis as well as some of our products from Genotypic. The floor is yours, Dr. Raja.
Yeah, thank you, Sri, and advance Independence Day wishes to all from India and Pakistan, Sri Lanka, almost everyone around. Everyone got independence almost the same time. So, a little bit more about Genotypic journey. We are now into the 25th year. We started as an R&D company, then we became a service company. Um, so they are all in very little things here. Illumina services, Ion Proton, Agilent, uh, microarray services, nanopore services. We also distribute nanopore products in India because it's more research oriented um, and a new technology. So our mission is actually simplify this complex genomics process because everyone thinks it's very difficult. I can't do it. Someone has to plan the experiment. Someone has to make the library. Someone has to sequence. Someone has to quality control and analyze. Someone has to publish. So we want to bridge this gap between this biology and the technologies, make it possible for everyone to do from beginning to end. That was our goal. Um, and we have a large enough team, few different companies, like what Sri said. Um, so today, what is that we are going to talk? First is what's Commander? Commander is the original name of Commander was Talaiva. In the name of Rajinikanth, we started the Commander. Later, we translated to English as Commander. So, how someone can do analysis without knowing command line scripting? That's what is Commander. And how to analyze data. See, there are new pipelines coming, new methods of analysis, new algorithms, new alignment tools, new base calling tools. So, unless you are an expert in bioinformatics and strongly follow this, you can never keep up with it, right? You can never keep up with it. Or sometimes you see in a publication there is a tool, and you don't know where to download it, how to run it, and things like that. And so this was one of the major problem in analysis. Unless someone has a team who's dedicated to analysis, one can't do NGS analysis. And apart from that, this compute power is another problem. Unless you have substantial amount of um, money spent on on servers, not just servers, it's hard. Um, so we are trying to bring the GT GPUs, the GPU compute powers, which will make available for small labs. So these two, we are trying to make sure that it will be a paradigm shift for people who are doing biology, how to do apply genomics, regardless of whatever be the system. So just to retell the problem, this is a million GB, 72 million GB of data generated in 2018. You'll be surprised, amongst which more than half is not fully well analyzed. And now we are getting into 1,000 million GBs. You calculate how much it is. I think this is several trillion, right? Several trillion, or rather 1,000 trillion uh, data, basis of data. I don't know when someone will analyze that. Always remained a challenge, and we are trying to break free of it and giving you a solution. So what is that? For example, if you're doing a 16S metagenome analysis or SARS-CoV-2, each one has different pipeline, right? But all of them has something very common. You have to quality control the data and also quantity control. Say if you say I'm doing SARS-CoV-2 sequencing, I need 100x coverage, right? So we have to see whether how much of data you have. If it's SARS-CoV-2, let's say 20 KB, so 100x should be 2 MB. So some data not filtering, not passing QC. So you'll already know how much of maybe 30, 40 MB, maybe 100 MB data if I have. It's good. And then you have to trim off both sides. You have to filter sequences which are below a quality score, for example. Then you have to align to the SARS-CoV-2 reference genome. Then you look at single nucleotide variants. Then you need to make it graphical output, then quality, then tertiary analysis. Then finally you make reports. You'll be surprised. This simple SARS-CoV-2, people take 24 hours to 48 hours. And a team of people working, bioinformaticians, postdocs working. But we made it much, much simpler. That's what you're going to hear today. See, the first part of analyzing data is command line scripting, right? Nobody wants it. I don't want it. I was forced to learn during Human Genome Project because nothing like Commander existed. Um, and if you go to online tools which developed later, how will you upload so much data? And then it has only one way of doing, like NCBI blast. Put all sequences, he will do only one thing. You can't do many, many things there. So there are now online cloud-based systems available. In all these cases, the problem is the same. Number one, you will pay a lot for these online tools. Second is you have to upload all the data, and you will still need a bioinformatician to do things. So let's hire a bioinformatician. Maybe some of you here listening are bioinformaticians. So let's hire a bioinformatician and do it. This is what most labs do. They will have one isolated bioinformatician. The poor chap, the guy or the girl, they are stuck with the biologist, number one. Number two is they never go to cutting edge unless there is a big team of bioinformaticians working. And they may not have the right servers to do. Some simple work might take days to do. Or find a collaborator. Many people say, okay, leave it. I'll find someone working in systems biology, someone in crystallography. I'll work with them. Maybe I'll share them the grant, share them authorship, and I'll work with them. Or some people go ahead and buy some NGS analysis software. It's kind of a black box. You don't know which algorithm they do, whatever it does, whatever it could do. That's also one option. If you're repeatedly doing something, you can buy a NGS analysis software. So now the question comes, who is going to analyze the data using, for example, someone says a GATK pipeline. Now you can't use a software, right? You can commercial software. So we are saying it's you. You can do the analysis yourself. So that's where we develop Commander. I'm going to show you very quickly for SARS-CoV-2 how it does. So be it Illumina, be it Nanopore, Iron Torrent, PacBio long reads or Nanopore long reads or MGI or any new things which are now coming out. All of them Commander can work. And you need not actually worry about, hey, I want to learn how it works for PacBio or MGI. Everything is clear, crystal clear, which I'm going to show you now. So this is in a way a tool, but this tool is something different. I don't know if you all watched any Rajinikanth Talaiva movie. So nothing is difficult, right? Nothing is difficult. Um, it's not essentially called a tool because this is going to run all the tools. A GATK pipeline, we didn't invent it, but Commander can run a GATK pipeline. Right? So de novo may be hard to install and get it going, but Commander will do that part. Okay. So Linux platform, Ubuntu version. Now we also have a Windows version. Though I don't recommend Windows version, but if you have a Windows computer and want to work on it, feel free to do it. Windows version is also possible. So major thing is you need not do any command line scripting. It uses only public domain software like GATK pipeline, NCBI blast. All of them you can do it on your computer. Of course, we have written some codes, written some QC tools and all, and it is you can customize it. You can visualize the results. Whether you do one sample or multiple sample, this can take care. Many of the public domain tools, you have to fire one job one by one by one. So this can fire for a whole folder. So how did we do it? So this Commander started with one time when Indian government said you have to have indigenous part to sell any software or any hardware into the country. That was around 2017 we started the Commander software. 2018 we started selling it. It was still some command line scripting required. By 2019 we had a full-fledged Commander, and here is when I think the COVID era was there. This is when the Commander was used extensively all around India, wherein is a person who just know RNA extraction can make a library, sequence it, that was using nanopore. And then the sequence data, whether it's from Illumina or MGI or come on, nanopore, you can sequence it, you can analyze it. Thermo Fisher, you can analyze, make a report, and upload. So quickly, I want to show how different Commander interfaces looks. This is the new one, which I'm going to show you now. So before you see the demo, quick demo I'm going to show is this is during the pandemic, people were all stuck with two places: shortage of technical laborers at different labs who can make library, run it. Second is analysis of data was became a big challenge. It became a really big challenge because many of them at that time, early time, was not cut out to work from home. So the bioinformatician has to come to lab, take the data in the hard drive, put it to the server, and then start doing analysis. So what we did was we we converted this whole process, we simplified this whole process and simplified this analysis challenge. No need for internet connectivity, and no need to upload anyway data to any of the servers which government of India banned it those days, and yet do everything and analyze COVID data. So this is one of the users, Dr. Krishna from Niri. Even three publications he has cited Commander. Sorry. Now let me now show you a small demo of Commander. So this is how the Commander, see here is the Commander logo. You click on it, the icon. So this is the new screen. This is how the Commander screen looks. And you can see, you can do a lot of things from FastQ to simple conversion of FastQ to FASTA that you can do. You can even process the variants. Maybe you got some 50 VCF files from different places. You want to put together and make a unique table. It's not adding the tables because it's more like a reference database, right? Demultiplex data, filter, trim, reads, almost everything you can do. So I'll quickly show you what and all it can do. See, for example, you can choose input path, which table it's in, which folder your data is sitting, or output, where it goes. You can import, export databases. You can add reference like a human reference or a SARS-CoV-2 reference. But anyway, Commander comes with everything pre-installed. We have human, SARS-CoV-2, all important ones are already sitting there. Rest of them you can import. And then different tools and workflows. See, tools means, for example, FastQ to FASTA format, right? VCF change. Just generate statistics. How much is the data which I got? How much is good data? How much is bad data? So these are all individual tools like filter reads. So all of them you can choose, create a project, or just choose an input file. You can do it. You can predict genes from a sequence. You have wide variety of tools, and you can merge and mix these tools. These are all standard tools, okay? Or you can go ahead here and choose workflows. So different workflows. This variant analysis workflow means you have a reference sequence, you have FASTQ files from different samples, you want to align to them, create a BAM file, look at SNPs, and then output a variant table. Or you want to output a consensus sequence of each one of the FastQ reads, it can do. So like that, different workflows, and you can create your own workflow, which is not present here. These are very commonly used workflows. So let me go and start this SARS-CoV-2 workflow, which we have been doing a lot, so that it's easy to understand. So as far as SARS-CoV-2 workflow is concerned, it's not a very complicated workflow. Someone has done sequencing. Obviously, they have done PCR of SARS-CoV-2, and then they have thrown it into one of those NGS machines. So you'll have reads, maybe millions of reads. Now you have to align to the SARS-CoV-2 reference and then create a BAM file, and then base call the BAM file, create variant table, and maybe translate those variants into amino acids, which gene, and then you also create a consensus table, go to some online websites to create what is this variant is called about. Is it a new variant? Is it a known variant, unknown variant? All of these takes a lot of time. Particularly if I give you 96 samples, it will take maybe 2 days for someone to make a report. So now I'm going to show you how it will take only maybe 2 minutes to make a report. So now I'm going to the input file. I'm going to choose this COVID data, and then I'm choosing this FastQ file, which has two barcodes. I can run as many barcodes as you want. And then I go to the toolbox. I want to see the SARS-CoV-2 analysis, and I say create project. So this project is created. So I have to go and see what is this platform. So this is Nanopore platform. See, I can choose PlaqueBio, Iron Torrent. Illumina, you can choose for Illumina/NGA also. Iron Torrent is each read will be still short, but the reads can be of 100 bases to even 500 bases long, right? So you can choose between what it is. I choose Nanopore, and then which workflow you want to run. So there is called Arctic pipeline. There is called general pipeline. General pipeline is the standard pipeline where you go to BAM because it will show you. Don't worry. What is the exact pipeline also you can see. It's not a black box. So we can choose regular general pipeline. How many CPUs you want to use? My computer has 8 CPUs, so I'll just put maybe 3 to use. And then there are different protocols. Arctic protocols or midnight Arctic, which means it's slightly longer ones. I choose that. I can save or set it as default. Once you set it as default, even if you have next, every time it comes, it will load this default and you can start running. So I set it as default, and then I have to just go and say run. So now he's already doing it. So I can see what's happening. So you can see below, he's just putting whatever he's doing. Okay. You can go down and see what he's doing. So he's creating, I'm just trying to, my mouse is a little bit. See, he has taken 2 sequences. Each one of them is, you can see he's merging all the FASTQ files, and then he's creating a SAM tool, he's creating bed file, he's going to variant call. So this he has not finished. So whatever he is doing, 100% you will know exactly what he is doing. So because my laptop is slow, two of this, it will take maybe 5 minutes to finish. And if it's a CPU computer, it would have finished this time 96 samples. That's the most interesting thing. Whereas in a CPU server, People will take at least 12 hours. When we actually did recently for a project, we took 2 hours sequencing run and 30 minutes analysis and gave the report. Morning samples came, before evening the report itself has gone, and people were surprised, how can you even make the report so fast? So this, now you can see while he's going, you can see what he has done. See, he's almost finishing work. Okay, you can see each process. See, he's creating consensus sequence for one of two is finished, so it's 50%. So he's creating a graphical, and he's going to the next clade. See, all from the data, instead of pasting it into a web browser and going, this downloads the Nextclade database. All the SARS-CoV-2 database is sitting in your computer, and he's analyzing it on that updated database. So your data is not going to the going online for analysis. See, now you can see he has finished one of them. See, some of them he finished for both, some of them he finished for one. So he is making all the reports. So you'll be shocked. The reports he is making. See, while he is making the reports, see, we can also go and see. I think he is almost done. He's almost done making the reports. See, you can also see while this is going on, actually, see, he has finished. So, so he has in 3 minutes, he has analyzed two SARS-CoV-2 sequences. So if you have 96, you can easily calculate. It will take 2 hours. On a GPU machine, the same time, 96 samples can be analyzed. I'll also talk about the GPU machine. Now let's go to the output and see how he is doing it. See here, output. For example, see now I'll go and see the output. See barcode 18. If you look at it, see he is creating a ready to go report. You can just convert into PDF and give it to anyone. For barcode 18, see, you can click and go there also. So a nice HTML report is done. You can save it as this thing also. How many reads are given? What is the maximum read length? What is the minimum? Each amplicon, right? There were, I think this contains totally 29 amplicons of around 1 KB. These are all overlapping amplicons. And then you can see this SARS-CoV genome, how much it's each amplicon, how much data has come, coverage has come, and what is the consensus, and what is the lineage it's predicted. It's a delta. So this lineage is predicted by few different methods. What is the genome coverage? How is the genome covered by X coverage depth? And then each variant, each position, what change to what, and if there is an amino acid change, it's giving. And this also saved as a table which you can again send it to people or look at it. And what type, this is annotation of all the different variants, each type of variant, how much you see. And then the S gene, what are the variants in the S gene, and each ORF. See, like this, he is going to create that for each one of the barcodes. And sometimes one need not go for each one of the barcodes, right? See this barcode 18, this barcode 17. Sometimes you only need a report. For example, if you look at consensus, each barcode is separate, and also all samples in one file also is given. If you look at here, this is the barcode 18, and then barcode 19 also will be here. See, I'm sorry, barcode 17. So everything comes in a single FASTA file. See, in this window it's opening, but otherwise it opens like a single file. That is a one. And then the variants, lineages, all given in a simple table. What is a barcode? What is a length X coverage? What is a genome coverage? And then
कोई ना यार
each.
टूटती रहती
Guys, can you please mute
Participants,
yourself?
kindly mute yourself, please.
So this table
We
also you can export, or you can just open this table in a Excel file also. So with this reports and this, so this is so quickly one can do it. People were amazed. For the same report someone has to do, I think there is two, three bioinformaticians are sitting to work to make one simple report. So the same thing can be done for many things, just some variant analysis, whole genome, 16S, exome analysis, whatever is commonly done can be done. And whichever way a report has to be made can be made. So you will be surprised that you do not need any bioinformatics knowledge, right? But you should know what you're doing, where you're going. I think that much everyone knows to complete a work. Actually, my son, so midnight sometimes we used to get the data, and then he fires the job, and then he takes, see, we always want to take a, for example, no, you go and open one of these files, open this, copy this file, go to the Pangolin database, copy paste, uh, and check whatever commander predicted is true in the online also. But you can do for just one or two and do it, or you can have a local blast also in commander possible. You can do it. So commander actually basically changed the way one, some of the our users said that, see, look, I have never gotten into bioinformatics or genome sequencing had I not seen commander. So we gave comfort to people, hey, buy this sequencer, buy any sequencer you want, though we prefer people buying nanopore, because nanopore itself is a simplified way of doing NGS, and then simply use commander. Then we found that people sometimes get stuck with the limitation of their own laptop or their own server, and then sometimes something else is running, there is not enough space, something not getting installed, all these kind of issues slowly were coming. That's when we started, let me go back to this slide. So this came in newspapers not too long ago. This is this year. How a low cost enable small laboratories to conduct genome study for virus and bacteria. This came recently. Um, so now comes the next question is, do you want to take 96 SARS-CoV-2 samples, put on a regular machine and wait for 2-3 hours? What will happen if you take 96 exomes, or if you want to assemble a genome? All this takes a lot of time. This is called as HPC, high performance computing. A regular server is not HPC. You should have a GPU. So that's where I think you, all the slides you would have seen, um, GPU, GTGPU, graphic processing unit. So do not spend too much time on analysis. So we have four different options. First is GTGPU laptop. You'll be surprised, this laptop is 15x faster than a normal laptop like one which I'm sitting, for not too much extra cost. Um, it can even do reference genome assembly, yeast, bacteria, and all one can do easily. Um, WGS analysis also can be done. Even some exome analysis can be done because a GPU is sitting there. And then we have GPU Mini, which is almost equivalent to this, but we have much more storage on this box, because many times after doing NGS, one of the major problem you face is, hey, where will I store this data? Sometimes copying from one hard disk to another hard disk itself is a problem. And then how will you store it? So these are, see, we give these machines which are all having storage which is mirrored within themselves, what is called as RAID. So even if one hard disk fails, the other hard disks will have the same data. So you will not lose the data. And then we have GPU Pro. This is a standard machine, can do lots of work. Okay. And then this GPU Tower, this is like a supercomputer. This is the highest version, but for most application, this version, GPU Pro is enough. So here is some example, comparison of a GPU with CPU servers which we have. Okay, you could see that the GPU can beat the. This is aligning short reads, 54 million short reads. I think this is for a panel, gene panel sequencing. You can see how GPU can outperform the CPU servers. So, what is the reason we created the GPU systems? We also created initially GT systems. Then we found that it's better to have a GPU systems, because one of the reason is we can test and validate everything. That's when the system comes itself is everything is pre-installed. So it's basically a plug and play system. And the system has data backup recovery, and we do the system monitoring maintenance and sitting from here. So who are the people who are using Commander or our systems? You will know ICMR, NIV have bioinformaticians, but still they prefer using our systems and Commander for certain applications. Some of them are companies like Zydus, Bayer, and RGCB is a large, and any, they have multiple sequencing systems. They use Commander, um, NIRI, which we showed before, for example. So last two slides, we are going there. So see, Commander is a paradigm shift. You don't need a bioinformatician. You don't need to do command line scripting. You might need a bioinformatician's help. I'm not saying they're not. This is like, why do you need to drive your bullet? You want to have a mechanic sitting behind you always, or you need him only once in a while. So this is basically a paradigm shift in allowing everyone to do NGS analysis. And GPU is a paradigm shift. This bringing high performance computer to anyone. Otherwise, previously you need a big air conditioned room, a rack servers, and high end IT guy to maintain those servers. No one will allow you to run them, someone else will run them. But this GPU is a paradigm shift. You can start with a small laptop or a mini system or a pro system, which is also not too expensive, or to a tower, which can beat any other. The tower which we have can beat some 7 or 8 CPU systems hands down, one single tower. So, folks, now these systems, GPU systems, and Commander is already being sold many places. Some 20 different countries have bought Commander. Is shipping around the globe. Firstly, it's pre-installed with all bioinformatics workflows. So basically, you're going to just plug and play, and we'll be there to train you also. So which means you're getting a box with Commander already installed, and once you start, we are going to log in from here to show you what to do initially. Though it's a very user friendly interface, one time we will run your analysis along with you. And Commander is just you can download from the website. There is a cost to it, but I think for those who are attending today, you can talk with Sri. His address coming next slide. You can get a short-term free license for you to do some analysis. So here, Commander is you start analyzing today, not yesterday. No, just you show the server, even your laptop, you install it and get going with it. Next level is you can customize with your own logos and names, and see now itself it gives dates and everything. But now we are putting small boxes where you write your name. So it will say analyzed by Dr. So and so or Miss so and so. And you want to put a logo and write something, it will put it on everything. And then it will also allow you to make your own pipelines. You say, I want GATK pipeline to do only alignment making until the BAM file. But I am having a tool which will do the create the VCF file. That's possible. You want to change the parameters on the VCF or even a different tool, everything is possible. So thank you everybody. Here is Commander. It's very easy. Commander at genotypic.co.in. You write to us. Even if you have some raw data you have not analyzed, or you have analyzed but you have a question, maybe you didn't analyze it well. Maybe you want to check it in a different pipeline. Maybe some experts want to look at it. We will look at it. Or you can send a WhatsApp message to Sri in this number, or of course, Genotypic. You're welcome to visit our website.